How microorganisms are discovered. In 1665, Robert Hooke, an Englishman, observed a thin slice of cork through a crude microscope and said life's smallest structural unit were little boxes or cells. But what he saw was just the cell wall, and it was the beginning of cell theory. In the late 1670s, Anton van Leeuwenhoek, who was actually a Dutch merchant and scientist, first observed live microorganisms and named it animalcules. He constructed more than 400 microscopes. He made some detailed drawings of microorganisms like bacteria and protozoa. In the second half of 19th century, many philosophers and scientists came up with a new theory called spontaneous generation. They believed maggots, which are larvae of flies, could arise from decaying corpses spontaneously, which means life could arise from non-living substances. But in 1668, Francesco Redi, an Italian physician also known as father of modern parasitology, experimented and demonstrated that maggots did not generate from non-living spontaneously. Then again, in 1745, John Needham, an English biologist, performed experiments and again claimed that microbes developed spontaneously from non-living, which supported concept of spontaneous generation theory. But after that, many scientists disproved this theory. Lazzaro Spallanzani, an Italian biologist, did experiments and disproved spontaneous generation and claimed life arises from pre-existing microbes. Then Theodor Schwann, Franz Schulz and Rudolf Virchow, they all disproved spontaneous generation, and Rudolf Virchow came up with a concept of biogenesis. Louis Pasteur, a French biologist, showed that microorganisms can be present in non-living materials, and new life actually arises from that pre-existing microbes. He also established germ theory of fermentation, germ theory of disease, immunization techniques, pasteurization techniques, etc. Thank you for watching this video. If you haven't subscribed to this channel yet, please subscribe and we will learn a lot of stuff together.